Hey everybody, today we're going to talk about transcription and translation. This is the process that cells go through in order to go from DNA to actually making proteins. And so transcription, we go from DNA to messenger RNA, and with translation, we go from that mRNA molecule to actually making proteins. All cells have the ability to go through translation because all cells have ribosomes. Transcription only happens in eukaryotes because they have a nucleus where prokaryotes do not. So let's start off with step number one, and that's transcription. In the DNA, in the nucleus, there is a code for specific traits, for specific genes. And so what will happen is there will be a trigger or some type of chemical message saying that you need a specific protein. And most proteins make up our genes. So what will happen is there will be a an enzyme called RNA polymerase that will come in and go to a certain section of the DNA. And what it will do is it will transcribe a message and create a strand that copies down the code, and that's where transcribe or transcription happens. So this little red molecule is messenger RNA. It will leave tiny little spaces in the nuclear envelope called nuclear pores. And so what will happen is the red will actually be able to leave the nucleus. And we talked about how the DNA does never want to leave the nucleus, it wants to stay put. And so the DNA stays, but that messenger RNA strand is actually able to move. And so that brings us to our next step, which is called translation. All right? And this is kind of a magnified version of a ribosome. They're actually really, really small. And so that messenger RNA will float and dock with that ribosome. And this starts the process of number 2, and this is called translation. So we're going from that messenger RNA to actually making the proteins that are needed. So on this red strand is a series of letters that carries with it a message. Each group of 3 letters is called a codon. All right, and so what will happen is there are different areas on the ribosome that will read these groups of 3. They're called EPA sites. We don't have to worry too much about that. What I want us to know is that they're read in groups of 3 letters. Now, what we haven't talked a lot about is how we actually create the protein. So what will happen is there is a molecule called tRNA, and it's called transfer RNA. And so what it will do is these groups of 3 letters are called codons. At the base of this transfer RNA, all right, also known as tRNA, and the T equals transfer, there will be what's called an anticodon. So it's the opposite of the codon. So the opposite of A is a U. The opposite of a U is an A, the opposite of a G is a C. So this is how the ribosome knows which amino acid to bring in. Is it uses the anticodon, it will tell the transfer RNA, hey, we need AUG, which is methionine. Each transfer RNA has an amino acid attached to it, so our first one here is methionine, which is MET. So what transfer RNA does is it will actually start here. It will read this codon. It will go and get methionine, and then it will bring it back to the ribosome. And there, it will then drop off MET, or whatever amino acid is needed. This red will then slide to the left, and we have the next codon that's red. So, transfer RNA will come in. Its anticodon will be CCC, since that is opposite of GGG. It knows that that amino acid is going to be glycine. So it will go and pick up glycine. And if you're wondering, well, where are these amino acids? They are created in the cytoplasm, or they're just kind of hanging out in the cytoplasm. So then, again, this transfer RNA, which anticodon is CCC, is then going to bring in the desired amino acid, which is glycine. So that's how this whole process takes place. First step, transcription. An enzyme will come in and figure out where that gene is, make a message. That message leaves the nuclear pores of the nucleus. That's all transcription is, DNA to making that red single-stranded RNA. Then the RNA is going to dock with the ribosome, which make proteins. Ribosome is going to read it in groups of 3 letters. Transfer RNA is really important because it is going to tell what amino acids to go and get, bring to the ribosome. This one was methionine, that's always the start. Then we slid down and it was GGG, so that we went to go and get glycine. And so what will end up happening is as this process continues on and on and on, it creates this string of beads. And those beads are actually proteins. Examples of proteins that are very common are keratin. That's what's in our hair and nails. Another one would be hemoglobin. That is in our red blood cells. One that is very fibrous and helps us look young would be collagen. We have some in our mitochondria that help us break down our food, rubisco. And so these are just some examples. Yes, they have fancy names, but they're found everywhere in biology and will help cells do a variety of things. So this is kind of our little breakdown of transcription and translation, and we'll be continuing to have labs and activities coming up this week. But we're now kind of expected to know how transcription and translation works. Have a good day, and thanks for listening.